Well, you can probably hear that I'm on the first day of a cold, so I am really sorry. And my two goals: one is to help you understand what I'm saying, and two is to not hack into this microphone. So, because that gets really awkward. So I'm going to try not to do that. Um, all right. So, like Pastor Tim said, my undergrad's in geology. I became a Christian while I was getting a geology degree, and at a state school at the University of Missouri. And a lot of my friends started asking me a lot of really hard questions. Like, they're like, well, that means you now believe that dinosaurs lived with people. I'm like, what? Like, I hadn't read the Bible. I had no idea, like, they knew more of the Bible than I did at the time. So a lot of the whole reason I got into science apologetics is, one, I wanted to have answers to my friends when they were asking me about how I had become born again. And then two, I just wanted to make sure I wasn't in a cult, honestly. So, because people are telling me that, and I'm like, well, that's a valid option, actually. So that's why I got into apologetics. It's always, I think, really good for people who don't agree to have really good questions, because it helps everybody grow. So, all right. So tonight I'm just talking about Darwinism and how Darwinism doesn't really make sense in the light of modern science. Okay. So first of all, I don't want to just talk bad about him, because there's a lot of good. Okay. So he was writing during the 1850s. I think up to Civil War times in America. He's in Victorian England. And he was actually quite brilliant, meticulous at data collection. I'm not saying Darwin's bad. Basically, Darwin knew what he knew in 1850s, and that's the way that science works. So I actually really like him, and I've been to his house. That's me in front of his house. And I would have pictures of inside, but the family still owns it, and they don't allow you to take photos. But this is the photo I got on the outside. But Darwin had a lot of inward struggles with 3 of his children dying. He was a super sensitive guy, and he was a great husband/cousin to his wife. So it was the 1850s. So if you want to read more about him and just get to know about his personal life and just him as a scientist, I really like, my favorite biographer of his is Keynes, or Kynes, Key. So I think it's like Keynes. And it's his great-great-grandson, who is a good writer, and he has access to all kinds of letters and history in the family. Anyway, so we're going to just focus on his theory for tonight. So it's important to understand that the actual main thrust of his argument had to do with something called independent creation or separate creations. So this is in a letter where he said that he's convinced that species are not immutable. So we now agree with him. But at the time, when he took natural theology class, they had adopted some Greek philosophies from Aristotle and things, where you just have this, where the creator made every single species independently. So he knew of lots of beetles. I don't think he knew of all 400,000 species that we know today, but there are a lot of them. And he was like, you know what? I don't think that each one of these beetles was created independently. Like, here's a green beetle, here's a red beetle, here's this beetle, that is what, when he wrote Origin of Species, in the introduction, this is what he opened up with, saying that he has no doubt that the view which he had been taught and had believed at one time, that species were independently created, is erroneous. He's like, I think that actually there's change going on, okay? I think that we can have some beetles changing into other beetles, okay? They're not just all independently created. And that was his main point was to overthrow the dogma of separate creations. And guess what? He did. Good job, Darwin. Okay, so this is the part he was right about. Okay, so I'm just gonna tell you his little argument, what he was right about, but then we'll go into the problems of how he then tried to extrapolate that into bigger changes. Okay, so in Origin of Species, his main argument starts with something that we all agree on: variation under domestication. Okay, and then he's like, well, if you can get change happening in animals by people breeding them, then you maybe that could be happening in nature, where just because of certain traits helping you survive, it's almost like nature's a breeder, and that would produce different forms. Okay, so he starts with domestication under variation. Variation under domestication, and then makes his way to nature doing it, okay? So he points out dog breeds or horse breeds, okay? I think everyone would be fine with that a wild wolf, then diversified into these domestic dog breeds, which are different variations, okay? And arguably, depending on your definition of species, some could be different species if they're no longer mating. Okay, if their differences make barriers to where they're no longer mating in the same gene pool. But he was actually not a dog breeder. He talked about that. He was a pigeon breeder. So what would not be that big of a deal for us today was kind of earth-shattering for him. So when he was had these different breeds, these specialized breeds of pigeons, and he would cross them to make more baby pigeons, when he kept putting them together, they'd start to look more and more like the wild rock pigeon. And we're like, yeah, duh. Okay, but why do we say, yeah, duh? Because he's the one who showed us that. He was the one who showed us actually not all variations within species are independently created, and maybe not even all species. So then he takes us from domesticated pigeons and dogs and moves his way into nature, okay? So I picked Darwin's finches because it's been in all our biology textbooks, pretty common. And basically, instead of having a breeder, like an actual person saying oh, I want a fast dog, or I want a hypoallergenic dog, or I want a hunting dog, or whatever, and picking out certain traits, it's the actual seasonal food that's available that's going to be influencing which beak is going to outlive other beaks of the finch, and that's how you're going to get different types of finches, okay? So all that Darwin helped us understand, and that we now have data for, is that different finches could all descend from one finch, okay? That different beetles, maybe all the different species of beetles descended from one beetle, okay? So, and everyone pretty much agrees on this, very much so. If you go to the Creation Museum or the Ark Encounter, has anybody been to the Ark Encounter? So you go in and you're so excited to oh, we're going to see animals, and you're like, they're not real, and they're weird looking. What are these funky looking animals in these cages? I've never seen these things before. Well, if you read all of the explanations, basically, it's the whole reason why they're saying that all the animals could fit on the ark is they didn't have to bring every single species on the ark. They just had to bring a representative from what's called a biblical kind that then after the flood would diversify into different species. Basically, they're using Darwin to say how they could fit animals on the ark, okay? So we need to give props to Darwin because he did collect a lot of data on that, and that's what we call microevolution, okay? So where you have small changes within a species Or even the ark is hoping, even getting whole new species from that. So that's where Darwin was right. But then he extrapolated that even further to say, well, what if these small changes that could lead to, new types of cats, what if that kept happening for millions of years and these small changes built up until you have a completely new animal, okay? What if the natural processes going on that are changing the finches population to have certain beak types, beak structures, or markings on their wings, what if that happened, those changes happened for millions of years, and then you would have a dinosaur, okay? So do you see how we're taking these small changes that we see and we know exist, okay, and we have data for? If you take an evolutionary analysis class in college, the whole textbook will be filled with numbers of these small-scale changes. But Darwin extrapolated it to say, and I think if these small changes keep happening, you can get whole new life forms. So it would make sense, go back in time, that everything just came from one common ancestor, which we call the senescester now, like our single-celled ancestor, and then it just started to diversifying into different body types through these changes, these microevolution changes, until you get all the life you see today. And that's not microevolution anymore, that's macroevolution, okay? So we're gonna look at why that doesn't make sense anymore. Now, could Darwin imagine that? Okay, could Darwin say, is that a good guess in 1859? Well, let's see. In 1859, they thought that the cell was just a blob of protoplasm. Okay, they didn't know about information, the DNA we talked about last week. He didn't know about molecular machines. At the time, they thought they were tiny people in sperm, okay? So they could think a lot of things. And he could imagine, okay, if these finches keep changing over millions of years, they can be something completely different. Okay, well, that doesn't make sense anymore. So because now we know about information, now we know that to get new body plans, you actually need whole new genes and whole new gene circuits, okay? So I'm gonna try to break down for you, what is it, how do we get, what's the mechanism, the process to get a new species, and then what would you need to have happen for new body plans, and you'll see they're completely different processes, okay? So, all right. So to get new species, or new variations, like we saw with the finches or beetles or the cat kind or whatever, you're gonna have already existing genes being shuffled around into different combinations, okay? So, but to get a new body plan, to have a finch evolve into a dinosaur, that's not just shuffling around bird genes to get different combinations of bird genes, okay? That's an entirely new suite of information and gene circuits. Could Darwin have known that? No, okay? But we do now, so we need to stop pretending this can happen. Okay, so I'm just gonna I'm doing 7th grade life science, okay? So don't check out on me. When my husband starts talking finance and economics, I'm like, I'm so not listening. Okay, but if he talks to me like I'm 5, one time when it came to our bills, he actually had to get out stuffed animals and make them talk for me to listen. I was just like, I do not care about money. So if you don't care about science, just I'm gonna do the simplest make it super simple, okay? So we're gonna do beetles because Darwin loved beetles, okay? And we're gonna look at this beetle gene pool, and we've got red, yellow, and orange beetles, okay? So they have 2 little letters on them. So these are called alleles. We're just gonna call them how genes are expressed, okay? And these letters code for their color. And they have 2 because you get one from your mom and one from your dad. So you get to give half of your chromosomes to your kids, okay? So if it's a red beetle, it has 2 big A's. If it's I can hardly see this. If it's orange, I think it has a big A and a little a. And if it's yellow, it has 2 little a's. And that's what codes on that gene for color to determine what color it is, okay? So in this population, when you have, generations on average, that's going to give you about 25% red, 25% yellow, and 50% orange beetles, all right? So this is called gene frequency. But what happens with something like genetic drift or microevolution, it can change the look of a population. So let's say that 2 little a's, so what color would that be if they're little a? Yes, good. So 2 yellows, they get separated from the beetle colony, and they can't get back, and they have to make their own new beetle civilization, okay? So when they go to have beetle babies, after several generations, are there going to be red and orange? Was the big A gene expression, was that even available to the yellow beetles? It's not inside of them, okay? So now the overall population is 100% yellow, okay? That's called change in gene frequency. And if you read an evolutionary analysis textbook, it's going to be filled with numbers like this. And it's going to be talking about population change and all these, bringing about new variations. And this is a really simple way to explain that it happens by shuffling around or different combinations of already existing letters, already existing code, okay? For that population to go from mostly red to all yellow, did new genes appear out of nowhere? No, okay? Darwin couldn't have known that. He didn't know about genes. He didn't know about information, okay? Another classic example of genetic drift in a textbook would be with these rainforest butterflies. So we've got orange and purple pigment in these butterflies. I know it's one butterfly, but it's supposed to be like a big population of them in the rainforest, and they code for orange and purple. But sadly, rainforest deforestation happens, and some get cut off from the original population, and it just happened that they didn't have as much of the orange alleles or the orange gene expression, that code for orange, okay? So they look more purple. And if they're separated and they're cut off from each other to make a new population for our textbook story, the new population is going to have even less orange, and so it's going to look all purple, okay? If that happens on the other side of the rainforest, on the other side of the original population, and there's not much purple available, they will start to look more orange, okay? So this is a textbook example of what's called a ring species, okay? And this is where you would learn in biology that there are now 2 different species because the purple and the orange no longer mate because of their markings, okay? Sometimes this is very simple with color that you can show, but it might be that maybe it was the code that has to do with mating season. Maybe they just now mate at different times, and so they're just not mating, so now they're a separate species, okay? So Darwin would see something like this and say, I think this purple butterfly and this orange butterfly came from the same I think they evolved from the same butterfly population. And we would say, yes, that's right. And he would say, maybe if that keeps happening, they will evolve into a totally new, into a praying mantis. I don't know, to a totally new insect, okay? Well, in the 1850s, that would be easy to imagine. But now we understand what was the mechanism for getting different colored butterflies. Was it getting new genes? No, it was actually losing genetic diversity. It was losing access to the original gene pool, okay? So I see people nodding heads. That's a teacher's dream. Yes. Okay, so that's genetic drift, where you're getting less genetic diversity, okay? You can also have gene frequency changes in a population by pools coming together, and that's going to change your overall gene frequency and the look of the population as well, okay? But there are already existing genes that were already in that type of animal. They're just going to be expressed in different percentages now. And then natural selection, as Darwin was correct, changes gene frequency also. So sticking with the beetle textbook scenario, certain beetles are going to blend in with the trees and others won't. So pretty soon, are there going to be many brown beetles left? Yes. I'm like, why is Joseph looking at me like that? Yes. Okay, well, are there going to be many green beetles left? No. Okay. Yeah. So they're going to get picked off, right? So, and does that remind you of anything you learned in school about moths? This is a classic natural selection example of peppered moths, okay? So most of us learn about this, where we have a major shift in gene frequency that was noticed because of natural selection, where the environment was changed. So peppered moths, there are darker ones, there's more peppered ones, and the darker ones, that they were pretty much almost like black colored, smoky colored, they would be eaten, but the peppered ones would blend in. But then after the Industrial Revolution, there's no EPA yet. You have all this soot and everything coating the tree trunks. Now who's going to stick out? The peppered one, and then the black one. So it went from like from one gene expression to the other. When I learned about this in school, I really thought, from what the teacher explained it, it was like the peppered moths were like, oh, we need to turn black, and they just morphed into black moths. That's not what happens, okay? So natural selection, all it's saying is that some gene expression helps you survive in some situations and others don't. But are those genes just appearing out of nowhere? No, they're what's already in the genome of the insect, all right? So hopefully, when now you understand that a change in gene frequency of just causing variation within a species of the same body plan is a completely different mechanism from what you would need to get a whole new body plan. You need whole new genes, okay? In fact, genetic drift, which I was always taught, that's a big driver in evolution. But genetic drift is actually a big concern to conservationists who, like pandas, that animals that are losing genetic diversity and they actually don't evolve through genetic drift, they actually enter an extinction vortex through inbreeding and they start to go extinct. So, all right, but Darwin couldn't have known that. So, real quick, the analogy that I like to use is cards. So like a deck of cards, that would be like the original created kind genome of an animal, okay? And then it just gets doled out into different hands. So like in poker, you start with the original deck, you deal out the cards, and then everyone has a totally different hand. Is anyone the same? No. So that's kind of like how you could think of how you have God creating the original cat, okay? And that weird looking thing at the ark, maybe that's what it'll look like, I don't know. But, and then through just many generations and different environments, different gene combinations that came out of that original deck are going to survive better in different environments, and that's how you get different species of cat. That's kind of like a easy way to thinking about. But if I say that to someone, you might have come across this if you were to say to someone, well, microevolution is just a change in gene frequency. But then usually the answer is, but millions of years. But it's millions of years. It's like, okay, well, if I deal out cards for millions of years, am I going to get new cards? No, I will get a ton of different combinations from those 52 cards. But if I want a whole new body plan, I need a whole new deck of cards, okay? All right. So a lot of evolutionary biologists are aware of this, and there's now a group called A Third Way. Basically, they're like, we want to still be evolutionists, but we don't want to be creationists, and we don't want to do intelligent design. So they're like, we're trying to find a third way. But this was a paper written by some of those guys saying that genetics might be adequate for explaining microevolution, but microevolutionary changes in gene frequency were not seen as able to turn a reptile into a mammal or convert a fish into an amphibian, because they need whole new genes. Microevolution looks at adaptations that concern the survival of the fittest, not the arrival of the fittest. The origin of species, Darwin's problem, remains unsolved. So that they kind of coined that term, survival of the fittest, but it doesn't help with the arrival of the fittest. Where do you have the original deck of cards? Where do you have the original code coming from? Okay? All right, that was problem 1. And you're like, how many problems are there? Okay, there'll be 4. But 2 of them are pretty short that don't require a lot of explaining. So problem number 2, I was taught that mutations are the raw material for evolution. So Darwin said that without some new profitable trait, natural selection can do nothing. He's like, there have to be changes for natural selection to act on. But then he says, of course, the laws of inheritance are quite unknown. He had no idea about Gregor Mendel. Actually, that's not true. On his desk, Gregor Mendel had sent his findings on pea plants in the beginnings of genetics. They were contemporaries, but I don't know if he read it. But he surely did not know about genetics. And so it was later when information was discovered in DNA, it was a good guess was, well, maybe this is how Darwin's change could happen. Maybe this code gets tweaked a little bit, and then that makes a whole new animal. Okay, that's a good guess. Honestly, if it worked, I would, people are like, well, you just don't believe in evolution because you're a Christian. No, I have no problem I have Christian friends who believe that God made evolution happen. If this was data driven, then I would be like, yeah, I believe in evolution. That's fine. But as of right now, the good guesses have no evidence for them. So mutations are supposed to, I was taught, make new functioning proteins. Okay, and before we talk about how I'm going to go back and explain, I'm not sure if you guys remember from high school, DNA to protein, okay? So you don't have, this is just I typed out, on the left, we've got our DNA base pairs, okay? And it's in the nucleus, and then a messenger comes. Is this jogging anybody's memory? A messenger is made, and then that message is sent to a machine called a ribosome. And that ribosome actually looks at every 3 letters as if it was a word and translates it into another molecule called an amino acid. And then all those amino acids are going to link up together. And if they're in the right order, then they fold into the correct shape. And then that shape works like a key in a lock. And it will start catalyzing reactions in the cell, okay? Or it's that perfect shape will be used in a molecular machine. But it has to be the perfect shape. Because, you order something from IKEA and a part comes and it's bent. Is that going to work? Are you like, oh, I'll just make something new out of this? No, you send it back. So the whole idea was that maybe if you have some DNA tweaked, kind of changes a little bit, then maybe that would line up a different sequence of amino acids, and then that would make the protein fold into a whole new shape, and it would do something new, okay? So as of right now, there are no mutations that produce new functioning proteins, okay? So this is what happens when you have a mutation. You end up with the wrong amino acid, so the ribosome reads the 3 letters and it's the wrong letter, or if it's a frame shift, then all of it's wrong and all the amino acids are wrong. And then I just picked one where only one amino acid is off, like in a point mutation. And you don't have a new functioning trait, you have a disorder, okay? So unfortunately, mutations only break meaningful code. They don't bring about new meaningful code. Do you remember what we need with the body plans? For new body plans, we need new functioning code, right? But that's only breaking it. But sometimes this is confusing because you'll say there are no helpful mutations. No, there are. Actually, they can be helpful. So a really easy example would be like the polar bear. And this is an example of also making a new species, not through getting new genes, but by breaking them, okay? So a polar bear and a grizzly bear can actually mate and make a grizzly bear. And they are pretty sure that a polar bear was just a brown bear who had a mutation and can no longer produce pigment, and so now it's just white, which happened to be good in the snow. So that worked out, right? So in that case, the mutation was helpful, okay? Or a textbook example would be if you have sickle cell anemia, you are resistant to malaria. So I was taught, see, that shows that mutations are giving you new traits, okay? But how does it make you resistant to malaria? Usually, the hemoglobin is nice and round and pillow-shaped to bind with oxygen and go around your bloodstream. But when you have a sickle cell, it's going to be sickle-shaped and it's all gooey and glommy. So when the malaria parasite gets into your red blood cells, if you have normal hemoglobin that's nice and pillow-shaped, it just kind of bounces off of the plasmodium, and the plasmodium just slowly kills you, okay? But if your sickle cell and that protein is misshaped and broken, and it's painful for you to carry oxygen, and you have a shorter lifespan, but it just so happens that broken hemoglobin will just go bleh and suffocate and kill the plasmodium, okay? So looking at that, that's a helpful mutation when it came to suffocating a plasmodium, but did that mutation give any new code or new meaning? No, it broke the code that happened to be helpful, just like in our polar bear situation. And so far, all helpful mutations are something like that, where they can be helpful in some situations, but we're more concerned with, are there mutations that bring about whole new genes to make whole new body plans? And so far, no, they do not exist. If they did, that would be super cool, like in X-Men or something. So Michael Behe, who wrote Edge of Evolution, talking about this, he's a biochemist, said random mutations much more easily debilitate genes than improve them, and that is true even of the helpful mutations, like we just said. So it's actually quite possible that a lot of evolutionary change is happening from degrading of meaning, and that's how you're getting differences, like even maybe within kinds. They don't know yet, but it's quite possible that big evolutionary changes, like from cat kind to different species, they know this is true in some dog breeds, are because of breaking information, where that's a completely different mechanism for if you want whole new animals, you need new information. Are you getting that? Am I repeating it enough? Okay. All right. All right, so in that point, mutations are typos, okay? They're not going to give you new meaningful code, unfortunately, okay? Problem number 3, usually I have just 3, but I have 4, because I added this one in not too long ago. Whenever I would talk with somebody and they would be telling me why evolution was a fact, and I would say, like, well, are you open to hearing like some issues with it? And I would always have these big 3, but then I added this one because this one's getting to be a really big problem. So, and these are orphan genes. So I started hearing about them just a bit in like 2009, and then there hasn't been a whole lot of explanation of where they could come from. But these are going to be any genes that when you're comparing species or similar species, and I'm talking species, not just like in the same family or the same phylum, like these are things that are close together. They actually find that there are these sets of genes in some species that are nothing like the genes in the other species that they supposedly descended from. And I'm going to be honest, this could even throw a wrench in the Ark Encounters theory on descent from a biblical kind, because they have a lot of change happening, but until they map all those genomes and make them public, I don't know. But anyway, so orphan genes don't show any detectable sequence similarities to genes in the genomes of other organisms. They constitute a large portion of every sequenced genome and are common among other arthropods. This is from a paper. Over 50% and 36% of the genes in the genome of the Lone Star tick and water flea, respectively, were identified as orphan genes. So all that's basically saying that, just let's look at the tick, that half of its genes aren't found in other arthropods similar to it. That's a really big problem for common descent. So, because the whole idea is you're supposed to have like this set of genes that were then mutated until you have a whole new animal, but you should be able to see that they're homologous, meaning that they share at least some code, but they're not homologous. They're totally different. And when you try to look up and research, like, how do they think how did this happen? Like, even one of the options is de novo. Like, they came out of nowhere. So that kind of looks like last week when we talked about finding out about the universe, and it's like, oh, it came out of nowhere. It's like, oh, what a trend. This also came out of nowhere. So orphan genes are found every time a new genome is sequenced. This is Richard Buggs from London. He's an evolutionary biologist. Their ubiquity has been one of the biggest surprises of genomics over the last 20 years. Many researchers had hypothesized the number of orphan genes found would steadily diminish as more and more genomes were sequenced, but this is not the case. Orphan genes continue to comprise a sizable proportion of each new genome sequenced. So it's, mapping whole genomes, or I guess people are saying genomes now, like we're going over to the British way, but I'm going to stay American. I'm saying genome. Okay, so anyway, but when they started mapping genomes and they noticed this and they were like, oh, I'm sure it'll go away, but it's gotten worse and worse the more that genomes they map, that they're actually they're not showing common descent. They're having big sections of genes that are not related to who they were supposed to evolve from. Does that make sense? All right. He goes on to say orphan genes are the hard problem for evolutionary genomics because we can't find other genes similar to them in other species. We can't build family trees for them. We can't hypothesize their gradual evolution. Instead, they seem to appear out of nowhere. So that's why I added it to one of the big problems, because back in when I started reading about it in '09, it was like, oh, we'll figure it out, and they haven't figured it out. So I've added it to my problem list. So there's a book that came out in 2020 by Dr. Tan and Rob Stadler, I think, and he's a biomedical engineer, and I think she's like a biochemist, but she did some postdoc in genetics. I think they met at Harvard. Anyway, it's a newer book, and they're not really affiliated with any movement, so that's why I thought it was really cool, where they basically were just taking computer programs. They're like, okay, we're taking all these genomes, we're mapping all of this DNA from all these organisms and putting them into a computer. And when I'm saying all of these organisms, we're talking about like bacteria, archaea, like unicellular things. And they're like, there's tons of their genomes in here. It shows all their DNA. Let's use the computer to compare them and see like how much overlap they have and see which genes are called universal genes, okay? So this is just with unicellular organisms, okay? And with prokaryotes, which means they don't even have a nucleus, okay? So taking the simplest of life, which for me, like, I mean, it's 2025, but I can still kind of imagine bacteria evolving into other bacteria. Like, that's not too hard for me. But anyway, but there's even a problem with that, where when it said that evolution predicted all organisms share the remnants of a core set of essential genes. However, the biochemist Dr. Tan reported in 2019, and then wrote a book in 2020, that across 1,000 prokaryotes, not a single protein code gene is universal. And that's just absolutely mind-blowing, considering what I taught was fact was as a kid. And now that they actually have the DNA, they're not seeing this evolutionary tree, a descent. They're actually, it's almost like they were created with totally different genes, okay? And that's why they've even had to split bacteria from archaea of unicellular organisms, because they actually have completely different information processing systems as well. So how they transcribe and make proteins is totally different. So then you have to explain how this unicellular organism evolved into another one, but then also evolved an entire different information processing system. So it's just getting pretty crazy and ridiculous, honestly. So, all right, so that was problem 3. Problem 4. All right, so problem 4 is the Cambrian explosion. And has anyone ever heard of the Cambrian explosion? Okay, good. Yes, this is an older one, oldie but a goodie, because even Darwin knew about that. And he basically said, if we don't figure this out, it's going to annihilate my theory, okay? Another thing I liked about Darwin is he was extremely intellectually honest. And personally, I think like in the 1950s when they discovered DNA, he would have been like, okay, yeah, never mind. This is not going to work. Like, he was very open. Like, he has an entire chapter in Origin of Species about, if you could discover this, then my theory won't work. If you discover this, my theory won't work. And one of them was, if you don't figure this issue out, it will annihilate my theory. So at the bottom of the fossil record, all of those body plans we've been talking about, like how can you get whole new body plans, all of them are fossilized at the bottom of the fossil record. And sometimes when I say that, it's confusing. My students are like, there's people down there? Like, no. So people are in the phylum chordates, okay? So basically all of the representatives of every animal phyla are down there, okay, at the bottom of the fossil record. I'm going to take a sip of my tea so I don't cough. All right, so we said that supposedly he was hoping that we would just keep digging and digging and digging and find the common ancestor of all of those body plans, of all of those different phyla, but that has not happened. the problem's gotten quite a bit worse, actually, because when below the Cambrian, there is the Precambrian, and there fossils can form there. They do find fossils of I think sponge embryos, things, small things that have been fossilized. But then right at the Cambrian, you have what's called the Big Bang of life, where out of nowhere, out of nowhere, you have fully formed body plans with no common ancestor that they all descended from, okay? So Darwin had predicted, and I was taught, that this is what the fossil record would look like, where you have at the top is going to be all of the different body plans, okay? I keep saying body plans and phyla. Think like echinoderms, like starfish. They're really different than cnidarians, like jellyfish, which are really different from worms, just different structures that require different gene circuits to make those different plans, okay? So he had guessed that they would all have come from a single ancestor, and if we kept digging, we would see evolution in real time through the fossil record. But after 150 years, Jonathan Wells says, we didn't find the tree of life, we found the lawn of life. It just kind of looks like grass. So you have body plans at the bottom of the fossil record. Some of them go extinct, but they still survive today. And there's no, you don't see a story of evolution or a common ancestor, okay? So just in summary, the problems that I went through, that shuffling pre-existing genes are not going to make new genes, okay? So we're going to have Q&A, so make sure that if you don't understand any of these, if I need to explain it better, let me know. Breaking code, that would be mutations, don't make new meaningful code, which would be whole new gene circuits and body plans, which macroevolution needs. Most types of animals have genes that are nothing like the genes of similar animals. That's the orphan genes, which is a growing problem. And then life's major body plans appear fully formed at the bottom of the fossil record without a common ancestor. So that is the Cambrian explosion. So those are the big four that I picked out. It takes me like a semester actually to teach everything, but I picked four that I thought would be helpful